Hello, everyone. This is Mark Plaster, Senior Executive Editor of Emergency Physicians Monthly, and you're listening to EP Talk, the place where emergency physicians around the world come to talk about things that are important to us, both professionally and sometimes personally. So today we're going to be talking with an expert from France. We're going to hear a little bit about a study that affects our everyday life in so many ways that oftentimes we forget. But first, I'd like to introduce you to Professor Oliver Mimoz from the University of Poitiers in France. Welcome, Professor. Nice to have you.
Okay. Thank you for this kind invitation, and it's a pleasure to be with you. So my name is Oliver Mimoz, and I'm working in the emergency department of the University Hospital of Poitiers, which is in the middle part of France. And my research topic is on prevention of healthcare-associated infection. And it's my pleasure to present you the results of the CLEAN3 trial.
This is the CLEAN3 trial, which was recently published in the Lancet Infectious Diseases journal, dealing with, Dr. Mimoz will explain the purpose of it, but basically it's a pretty straightforward issue. It has to do with infections in IVs, and there are billions of, I don't know the exact number, but there are over the background information said over 2 billion IVs are placed annually worldwide. And we all know that. It's reflex in the emergency department. Somebody comes in and they just reflexively get an IV for whatever reason. And sometimes that is associated with secondary infection. And if anytime we can prevent that, that is to their advantage and certainly to our advantage. Hospital infections are a huge issue. And it's so this, even though it seems like a pretty simple, straightforward study, this is one that affects us literally every day of our professional lives. So I just wanted to jump in there and kind of frame this and then ask Dr. Mimoz to tell us a little bit about the CLEAN3 study.
So, yes, you are totally right. Sorry. The peripheral intravenous catheter is the first medical devices used in the hospital. And in my country, we sold about 3 million catheters per year. And if you look at the world population, more than 2 billion of these catheters are sold each year. So it's the first medical device used. And usually it's called as a very simple and safe device. But unfortunately, it's not. You discuss about complications infection, but we also have non-infectious complications. And we know today that about 1/3 to 50% of those devices fail before the intended dwell time. And most
I think, Professor, that's actually worth repeating because you're absolutely right. We think about infections in the peripheral IVs and the catheters as being a problem, but also for the patient. You know, if you get an IV, I mean, I recently had one, and if you get an IV and you want it to be able to stay for your period of time in the ER or in the hospital, whatever it takes, and if that peripheral IV fails before its, in essence, what I would call its due date or its potential date of living because of potential infection or other things like that, then you created not only an inconvenience for the patient and potential danger for the patient, but you've also created additional cost and for the hospital and some other things. So it's not just, it is important to stop infection, okay? But it's also important to prolong the life of the peripheral IV catheter. So anyway, I just want to jump in there and reframe that for you.
Yeah, and this complication also occur early during the course of the catheter. Most of the time, during the first 2 days after catheter insertion. So as you said, not only it's a complication for the patient, is inconvenient for the patient, but this is also costly for the hospital. So this is why we need to develop new approaches to reduce infectious and non-infectious complications related with the use of those catheters.
Okay,
and
great.
this is the aim of the CLEAN3 trial. The CLEAN3 trial was developed to propose new technologies, new approaches to reduce infectious and non-infectious complications related with the use of this kind of catheters.
Absolutely. Very simple. I like studies that have a very simple endpoint. You know, does it prolong the catheter? Does it reduce the number of infections and colonization? That's an important. I noticed that that was one of the things that was most striking was catheter colonization. We don't think of the bacteria that are growing inside of the catheter, but there are. And so that is a source of seeding of bacteria for the patient. And in today's hospitalizations, patients are very susceptible to that. And
and we also reduce other complications such as catheter occlusion, catheter displacement, every cause of catheter failure. And the problem with catheter failure is we have to stop a patient's treatment, which may be deleterious for the patient, and we have to replace a new catheter. And as you know, replacement of a new catheter is associated with pain to the patient, and cost for the hospital. So this is why we have to fight against this kind of complications, which is very important, especially because this kind of catheter are frequently used in hospitals.
So the CLEAN3 trial was not small. It had tell me about this. It had 1,000 patients in 9 different medical wards within the single hospital of is it Poitiers University Hospital?
Yeah, absolutely, yes.
Tell me about it.
Yeah. It's 1,000 catheters in 9 medical wards. It's a study conducted during 6 months, so it's a relatively big one. And the study was set to be able to answer to 2 major questions. The first one is how to prevent catheter colonization, which kind of antiseptic we have to use, which is the best one, and we compare the two products which are most used in the hospital with the alcoholic formulation of chlorhexidine to an alcoholic formulation of povidone iodine, and we also compare 2 kind of devices, a standard approach to a combination of innovative solution. Against to be able to determine which one is the best for the patient.
So just to recap, it's not just looking at the different solutions. It's comparing alcohol and iodine, povidone iodine, against alcohol and chlorhexidine, but it's also looking at the actual apparatus of the IV. We sort of gotten used to the standard catheter hookups, and you're looking at a different system there, trying to see if that different system also provided for safety. So what were the results of that?
We have two kind of results. Comparing antiseptic solution, we demonstrate that the use of the alcoholic chlorhexidine formulation used in a single-dose applicator was associated with a 92% relative reduction in the risk of infectious complication compared with the use of the 5% alcoholic povidone iodine solution. And related to catheter failure, we also observe that the use of the bundle of innovative solution was associated with a 27% reduction in the relative risk of catheter failure compared with the standard approaches. So we can conclude that the both approaches, the antiseptic solution and the combination of innovative solution, may improve the quality of patient's care.
So it's a pretty straightforward kind of assertion, or not assertion, but conclusion that you arrived at, is that the combination of using a different cleansing chemical on the skin, chlorhexidine, alcohol and chlorhexidine, and the use of a combination of an integrated PIVC and sterile needle displacement, needle-free connector, and a disinfecting cap. That's the combination you used on one side, and the other side was just simply a standard straight safety PIVC with a winged extension set to a three-way stopcock. So both IVs were continuously infused with saline or other polyionic solutions. So they were very similar, except for those specific things. And why do you think that? Particularly, I was interested to know in the apparatus, the integrated PIVC, and a positive displacement needle-free connector and a disinfecting cap. Why do you think that seemed to work? Do you have any ideas on that?
Yes, we before starting the study, we have very few information in the medical literature indicating that this kind of devices may be helpful in preventing complication when they are applied to patients. However, before we start with the CLEAN3 trial, this kind of information was not totally strong to recommend this kind of medical devices to be used in hospitals. So what we know is that this kind of devices have the capacity to reduce all the complications that we can observe with peripheral IV. Positive displacement needle-free, for example, connectors are able to reduce catheter occlusion. Disinfective caps have the ability to reduce catheter infectious complication, etc., etc., etc. So I think that when we use all these kind of devices in combination, we can reduce different complication, not only infectious but also non-infectious complication, and this is probably why we observe that by using this kind of material, we can prolong the catheter dwell time without occurrence of a complication.
It's interesting to note. We've spoken of this at the beginning, but it's interesting to reiterate that as big a problem as it is to have a blood infection from the IV, as big a problem is simply having to change it. It stops treatment. Sometimes it requires additional personnel to come in to place the IV. There are delays in treatment. How many times do we see staff being tied up with IV failures? And so anytime that you can streamline that, make that less invasive, less dangerous to the patient, then you're going to end up saving time, saving money, and saving suffering on the part of the patient. So that's an amazing study. I think, to be honest with you, I'd like to go on and continue to talk about this, but I think you pretty well stated the issue. Do you have anything else that you'd like to add to this?
No, I think that the next step will be to conduct a medical economic study just to demonstrate that not only we can reduce complication, but also our approach does not increase the cost of the solution. So this study will be done in the few weeks, and I hope that we will have results and a new publication before the end of the year. But I think that for my colleagues should be very important to demonstrate not only that we are effective, but we are also not. We do not need to increase the cost because this kind of approaches are cost effective. So that we have benefit without increased cost. So we are safer without needing more money. I think it's a very important message to my colleagues.
Yes, absolutely. In a time where we're moving more and more to pay for value, as they say, in which your goal is to not only provide better care, but at lower cost, at least cost neutral. But if you can get it at lower cost, those are definitely the kinds of endpoints that we're looking for. And especially in something that is like this, where even a minor improvement and this is very significant, the improvement in length of the catheter life and the diminishment of infection. I mean, those are significant. But when you have even minor improvements, and that happens over 1 billion times, the improvement is multiplied tremendously. So this is concentrating on a very simple but a oft-repeated procedure that if you can show a minor improvement in it, it will be magnified over 1 billion times, which is huge. And as you said, the same is true about the cost. If we can show that there is a better care at a lower cost and magnify that 1 billion times, then we're going to really see significant changes due to a very, very simple improvement of a common device. So this is I thank you so much, Professor, for joining me today to talk about this important yet simple issue that we deal with on a daily basis. And these are the kinds of things that emergency physicians like to hear about. So, Professor, thank you so much for joining Emergency Physicians Monthly, this version of EP Talk. I hope our listeners have enjoyed it. And join us again, EP Talk, where emergency physicians come to talk about things that are important to us. Thank you very much.
With pleasure. Thank you. Bye-bye.